There is a small, climate-controlled case in the Silkeborg Museum in central Denmark that is kept at a steady, cool temperature and carefully regulated humidity. Inside, it lies a man. His skin is the color of burnished leather, drawn tight over the bones of a face that has held the same expression for nearly 24 centuries. His eyes are closed. A faint, almost private smile rests on his lips. A day's worth of stubble dots his chin. Around his neck, cinched tight, is a braided leather cord, its long end trailing behind him like a tail. For 75 years, scientists have taken small pieces of him. A sliver of bone here, a scraping from his gut, a fragment of tissue. For decades, his DNA refused to answer. The bog that had preserved his face had destroyed his genome until recently. And the answer has made him a stranger. In the spring of 1950, on the mist-shrouded Jutland Peninsula of Denmark, in a stretch of bog called the Bjeld Skovdal, two brothers named Viggo and Emil Højgaard were cutting peat with their family. For centuries, this dense, dark earth had been the local fuel, a source of warmth against the damp Scandinavian chill. Then one afternoon, the work came to an abrupt halt. The blade struck something that wasn't peat. It was soft yet firm. As they cleared the surrounding earth, a human face emerged from the darkness. The man was lying on his side, curled as if in deep sleep. His features were astonishingly preserved. The family was stunned. They believed they had stumbled onto the victim of a recent murder, a body hidden in the bog by someone who had not expected it to be found. They contacted the police in the nearby town of Silkeborg. The police came. The archaeologists from Aarhus University came soon after, and within hours, the case shifted out of the realm of crime and into something much older. The skin, the cord, the position of the body, the depth at which he lay. None of it belonged to the 20th century. The investigators were looking at a face from the Iron Age. Radiocarbon dating performed years later would place his death between 405 and 380 BC. He was a contemporary of Socrates. He was alive in a world on which the Roman Empire had not yet pressed its weight. A northern Europe still entirely outside the Mediterranean's reach. The man pulled from the peat that afternoon had been waiting in the dark for nearly 24 centuries. Forensic details accumulated quickly. He stood 5'3" tall. He was between 30 and 40 years old at the time of his death. A respectable lifespan for the era. His wisdom teeth had erupted. His hands and feet were surprisingly soft, not the calloused extremities of a man who had spent his life behind a plow or hauling timber, which suggested he may have held some special standing in his community. He wore only a pointed cap of sheepskin, fastened beneath the chin, and a smooth leather belt around his waist. He was otherwise naked. The stubble on his chin indicated he had shaved a day or two before he died, and around his neck, the cord, a braided leather noose, 2 meters long, still cinched tight against his throat. The marks on his skin and the state of his cervical vertebrae left no honest room for doubt. He had been hanged. For 75 years, his face has been the most studied human face from Iron Age Northern Europe. Reproductions of it sit in textbooks across the continent. But until very recently, the body that wore that face was a closed door. His genome would not speak. The reason has to do with the chemistry of the place that preserved him. Bogs are extraordinary environments. They are waterlogged, anoxic and acidic, with pH values that often fall between 3 and 4, closer to vinegar than to ordinary groundwater. The primary component of the peat is sphagnan moss. As the moss dies and decays in the waterlogged dark, it releases a carbohydrate polymer called sphagnan. Sphagnan binds to nitrogen in surrounding tissue, locks up the calcium that bacteria need to function, and combines with the bog's high concentration of tannins to create an environment hostile to almost everything that causes a body to rot. In essence, the bog pickles whatever falls into it. The acidic water tans skin into leather. The lack of oxygen prevents the soft tissues from breaking down. Hair stays attached. Fingernails stay attached. The brain, though shrunken, often remains inside the skull. That is why Tollund Man's face survived. It is also why his DNA did not. The same acidity that cured his skin shredded his genome. Ancient DNA is fragile to begin with. Over thousands of years, it breaks into shorter and shorter fragments, accumulating chemical damage that confuses the machines that try to read it. Submerge those fragments in acid for 2.5 millennia, and what comes out the other end is an unreadable mess. For decades, every attempt to extract usable nuclear DNA from a northern European bog body had run into the same wall. The strands were too short, too damaged, and too contaminated by the modern hands and modern microbes that had touched the samples since exhumation. This was not a unique problem for Tollund Man. Across northern Europe, hundreds of bog bodies sit in museums with the same paradox written into their cells. Bloodline Man in Denmark, Tollund Man in England, and Eyed Girl in the Netherlands. Their faces tell one story with extraordinary clarity. Their genomes tell none. For the first half-century after his discovery, anything anyone said about Tollund Man's ancestry was an inference drawn from the period and the place where he died, not from the man himself. Then the instruments changed. The development of hybridization capture techniques in the 2010s allowed researchers to fish out the few intact fragments of human DNA from samples drowning in bacterial and environmental contamination. The cost of deep coverage sequencing fell by orders of magnitude. The use of the petrous bone, the dense pyramid of bone that houses the inner ear, became standard practice because it preserves DNA at concentrations far higher than other skeletal elements. The previous generation of instruments could not ask the question. The new generation could. The question, when it was finally asked, was not only who he was, it was where his people had come from. The answer, when it came, did not match what anyone had expected. The breakthrough came from his petrous bone. Researchers drilled a small sample from the dense pyramid behind his inner ear, the part of the human skeleton that holds on to DNA longer and more reliably than any other. They paired it with material from his femur. After repeated rounds of extraction, library preparation, and hybridization capture, they had something the previous decades had not produced. They had a sequenced genome, not a perfect one. The coverage was thinner than what modern, ancient DNA labs prefer, the gaps frequent, and the contamination higher than ideal. But for the first time, the man in the case had a readable genetic identity. The expected result was straightforward. By the late Iron Age, the population of Northern Europe was a settled three-way mixture, a base of Western hunter-gatherer ancestry, the deep substrate left by the people who had walked into Europe at the end of the last ice age, a layer of Anatolian farmer ancestry brought in during the Neolithic expansion that began around 7000 BC, and a substantial overlay of steppe ancestry carried west by the Yamnaya and their descendants in the 3rd millennium BC. By 400 BC, every Iron Age individual, sequenced from Denmark, Sweden, northern Germany, and the surrounding regions had carried this mixture in broadly similar proportions. Tollund Man was expected to fit the same profile. Broadly, he did. The three ancestral streams were all present. The autosomal genome placed him recognizably within the Northern European Iron Age. He was not, on first reading, a foreigner, but the proportions were not quite right. His steppe-derived ancestry was lower than the regional average for the period. His Anatolian farmer signal was correspondingly higher than what had been measured in the small but growing comparative data set of Iron Age Jutlanders. His Western hunter-gatherer fraction sat at the upper end of the local distribution. None of these deviations were extreme on its own. Taken together, they suggested a man whose ancestry was tilted slightly toward an earlier, less steppe-heavy version of Northern European demography than his contemporaries. The comparative samples mattered. By the time the Tollund Man sequence was produced, geneticists had data from Haraldskær woman recovered from another Danish bog. They had data from Iron Age cemetery populations in Jutland and southern Scandinavia. They had high-coverage genomes from individuals living within a few centuries of him in roughly the same landscape. Against this backdrop, Tollund Man was not an outlier in any dramatic sense. He was a quiet anomaly, a man who fit the neighborhood but did not quite fit the average. That was where things stood when the first analyses were published. A late Iron Age Dane with a slightly archaic-looking ancestry profile, carrying the broad genetic signature of his region, with a few unexplained tilts. It was a tidy story. It did not last. The deeper the researchers looked, the stranger the picture became. The deeper look meant zooming in on the parts of the genome that do not reshuffle every generation. Autosomal DNA averages out across centuries. A man's 22 pairs of non-sex chromosomes record the blended average of all his ancestors, smoothed and mixed by the recombination of every generation. But two stretches of the genome refuse this smoothing. The Y chromosome, passed from father to son, travels down a single male line virtually unchanged. The mitochondrial DNA, passed from mother to child, travels down a single female line in the same way. They are the genetic equivalent of surnames. They tell you not where a person's ancestors are from on average, but specifically which father begat which son and which mother bore which daughter, century after century. By the late Iron Age in Jutland, the dominant paternal lineage was the one that had been spreading across northern Europe since the Bronze Age. The R1b and I1 haplogroups in their northern subvariants accounted for the great majority of sequenced Iron Age men in Denmark and southern Scandinavia. These were the patrol lines of the steppe-descended population that had reshaped the region 2,000 years before Tollund Man drew breath. The mitochondrial picture was more varied, dominated by H, U, and K subclades that had been resident in northern Europe for millennia. Tollund Man's lineages did not sit comfortably inside this picture. His paternal line, on the deeper resolution permitted by the new sequence, traced to a sublineage that has been found mostly in older pre-Iron Age contexts further south, with only sparse representation in Iron Age Scandinavia. His maternal line carried a subclade rare in the Northern European record of his period, with its closest matches scattered across Neolithic and Bronze Age sites in Central Europe and the Eastern Mediterranean basin. In other words, his autosomal genome made him a Jutlander. His Y chromosome and his mitochondrial DNA made him something else. On the lines that act like genealogical surnames, his neighbors were not his cousins. The paternal line that should have placed him among the dominant local clans placed him instead among a thinner, older, more southerly current. The maternal line that should have anchored him to a regional founder mother anchored him to a tradition his contemporaries did not seem to share. The cousins may exist somewhere in a museum drawer in Hamburg or Bologna or Athens, waiting to be sequenced. The comparative library for Iron Age Northern Europe is still thin. Hundreds of individuals have been published, but hundreds are not enough to map the lineage structure of a continent. The cousins may also not exist in any recovered form. Tollund Man's family, on his direct lines, may have been small, mobile, or unlucky in the way they buried their dead. The man whose face became the face of Iron Age Europe carries surnames that do not place him squarely within Iron Age Europe. He looks like his neighbors. He does not descend from them. There is a piece of evidence that has been known for decades and that the genetic strangeness now forces us to read again. It is the manner of his death and the manner of his burial. The cord around his neck and the state of his vertebrae confirm without ambiguity that he died by hanging. But the body in the bog is not the body of a man dragged to the gallows and discarded. After he died, someone laid him on his side in the peat with care. His eyes were closed, his mouth was shut, his legs were drawn up as if in sleep. The pointed sheepskin cap was secured under his chin. These are not the gestures one offers to a despised criminal. They are gestures of reverence. The Roman historian Tacitus, writing 4 centuries later about the Germanic tribes to his north, noted that they hanged traitors and pressed criminals into bogs. The line has been quoted often in connection with Tollund Man, but Tacitus was writing about a different region and a later period, and the bog burials of northern Europe long predate his account. The pattern across hundreds of these bodies, scattered from Ireland to the Baltic, is not the pattern of routine execution. It is the pattern of deposition. Precious swords, ornate jewelry, cauldrons, pottery filled with food, and human beings placed with the same intent. His last meal sharpens the question. His gut contained a porridge of barley and flax, mixed with the seeds of more than a dozen wild plants, including pale persicaria and gold of pleasure, and traces of fish. The variety of wild seeds is unusual for the period. Some scholars read it as a ritual meal prepared for a person being given to the bog. Unlike other bog bodies, his gut contained no ergot, no henbane, and none of the psychoactive compounds that would have dulled what was about to happen to him. His mind was clear as he walked to the rope. The genetic finding does not solve any of this. It thickens it. In some Iron Age societies, sacrificial victims were chosen specifically from outside the everyday community. Outsiders, captives, and individuals marked by some form of difference were thought to make the most powerful offerings because they belonged less to the living world and could be more cleanly given to the other one. The temptation is to read Tollund Man's lineage anomaly through that lens. A man whose lineage did not match his neighbor's and was given to the gods. The temptation should be resisted. The careful arrangement of the body, the cap secured beneath the chin, and the closed eyes all argue against him being a despised outsider. He was offered, not discarded. Whatever marked him as special enough to be given to the bog, the genetics cannot yet tell us. What the genetics can tell us is that something did. There is a thing that happens with Tollund Man every couple of decades that is worth naming. A new technique emerges, a new dating method, a new imaging protocol, a new way of pulling information from tissue that previously refused to give it up. The scientists go back to the body in Silkeborg, take the smallest possible sample, and ask a question the body could not previously answer. And every time, the answer makes him less familiar than he was before. In 1950, he was a recent murder victim pulled from the peat by 2 brothers who thought they had found a hidden crime. Within hours, he was an Iron Age man, hanged, probably executed for some offense the period had reasons for. In the 1960s, refined radiocarbon dating placed him in the late pre-Roman Iron Age and made him a contemporary of Socrates. In the 1970s and 1980s, the first detailed analyses of his gut contents established the porridge, the wild seeds, the absence of any final meal of meat, and the picture of a meal eaten 12 to 24 hours before he died. The 2000s brought high-resolution scans, second-generation gut studies, and the slow accumulation of forensic detail that turned him from a face into a body with a medical history. Through the 2010s, repeated attempts to extract usable DNA from his tissues failed or produced fragments too short and too contaminated to read. The bog had taken his genome from him, or so it seemed. The early 2020s brought partial successes, refined methods, and the first comparative genomes from other Iron Age individuals in Jutland and southern Scandinavia. And then the petrous bone gave up its sequence, and the autosomal profile placed him recognizably among his neighbors. And the deeper look at his Y chromosome and mitochondrial DNA placed him somewhere else entirely. The direction of travel is suggestive. Every resolution, every improvement in the tools has made him harder to place, not easier. We keep thinking we have figured out who he was, and we keep finding that we had only figured out what our instruments were then capable of saying. The next round of techniques is already on the horizon. Ancient proteomics, which can read the protein signatures of tissues whose DNA has been destroyed. Single-cell ancient transcriptomics, which can, pickled, reconstruct which genes were active in which cells at the moment of death. And fine-resolution isotopic studies that can trace not just where a person grew up, but where they spent each season of their final years. In a decade, the picture we have of him in May 2026 will look partly wrong in its own turn. This is a useful thing to remember when reading confident assertions about the lives of prehistoric people. What we know is less a portrait than a series of photographs, each one slightly contradicting the last, each one taken with a better camera. For now, in the climate-controlled case at the Silkeborg Museum, Tollund Man lies exactly as he was placed, curled on his side, the cord still around his neck, the slight smile still resting on his lips. The tourists file past in respectful silence. Most of them are told in the exhibit text that he was a late Iron Age man hanged in a bog in central Jutland around 400 BC, probably as a sacrifice to the gods of his people. The text is true. It is also already partly out of date. 2,400 years after the rope went tight, he is still patiently managing to be a stranger. He is not done. The body in the case is not done. The mystery of his DNA just got way stranger. And if the last 75 years are any guide, it is going to keep getting stranger. Please like and subscribe for more such videos. I will see you in the next one.